We're back. Concept 32.3 says animals can be characterized by body plans. And despite the fact that there's a huge variety of animals, different types, different body sizes and shapes, most animals can be characterized by relatively few body plans. Zoologists sometimes categorize animals according to what they call a body plan, which is a set of morphological and developmental traits that kind of describes the creatures. And although species do vary tremendously in morphology, the diversity can be packaged or categorized by a relatively small number of major body plans. Some developmental characteristics are conservative. In other words, they have been around for a long time. They occurred early and they have hung around through countless generations of evolved species. For example, the molecular control of gastrulation, which is the infolding of that blastula that we talked about, is conserved among many diverse animal groups. So whether it is something like a human being or something as different as some sort of fish or a parakeet or something else, most animals go through this gastrulation process. So that process is what we call conserved. It began in our early ancestors and has continued to the present day. One of the aspects of body plan that we can look at in animals is called symmetry, and animals can be categorized according to the symmetry of their bodies or the lack of symmetry. One very basic basal animal we're going to talk about in particular in the next chapter, sponges. For example, many sponges have no symmetry. There's no looking at their body and saying, oh, well, this is the left or this is the right or anything like that. Some animals have what we call radial symmetry with no front, back, left, or right. And if you look at an example here like a flower pot, that's a good example of something having radial symmetry. A good example of an animal that would have radial symmetry would be, say for example, a sea anemone. So you can see the sea anemone you could cut the same way. Any imaginary slice through the central axis divides the animal into two mirror images. So you could cut the flower pot, say, this way, or you could cut it this way, and any slice through the center would give you two equal halves basically. So that's radial symmetry, kind of a round symmetry, radial symmetry. And some creatures, especially some of the more or the less complex, the simple creatures like a sea anemone have that. And then of course some of the most simple creatures like sponges have no symmetry at all. Two-sided symmetry is called bilateral symmetry, and this is what you generally see in some of the more complex creatures. Bilateral symmetry animals have a dorsal, which is top side, and a ventral, which would be, say, you could consider it the stomach or bottom side, a right and a left side. They have an anterior, which is the head, and a posterior, which tends to be the tail, and they have what's called cephalization, which is usually the development of some sort of head structure. So as opposed to, say, a sea anemone or a jellyfish that has no true head and doesn't have bilateral symmetry, you could look at, say, a lobster or a dog, and you can see that it has a dorsal and a ventral side, a right and a left, anterior, posterior, and certainly it has a head, cephalization. So that would be bilateral symmetry. And cephalization, that word, the development of a head, comes from the Greek kepel, which means head. In bilateral symmetry, like what you see here, they've used the little shovel as an example. So I said flower pot, but I guess it's supposed to be like perhaps a sand a bucket for your sand. But here the little sand shovel, when they show bilateral symmetry, like with the lobster, only one imaginary line, only one imaginary cut divides the animal into mirror image halves. Like you can only get two mirror images of the lobster if you cut it in this one direction. If you were to cut it in this direction, you would have two halves, but you wouldn't have two mirror images. So bilateral symmetry means there's only one way to cut it to get two mirror image halves. So here's a comparison of the two types of symmetry, or you could have no symmetry at all, which is somewhat rare in animals. But radial symmetry, say for example, it doesn't matter how you cut it, but one cut through the middle will give you mirror images of each other. And then bilateral symmetry, which is basically where you can divide only one way to get a mirror image. And bilateral symmetry also includes things like having a notable top portion and bottom portion, front and back and head and things like that. So some of those terminologies that you have probably heard quite a bit, you might want to make sure that you do know, such as dorsal, so think of like a dorsal fin of a dolphin or a shark, and ventral, and then anterior toward the head, posterior toward the tail, and cephalization, which is the development of a head. Animals that have radial division, radial animals are often sessile. Sessile means that they are either attached to a substrate or they're non-moving. So radial animals such as coral, or maybe an animal that has no particular symmetry at all, such as a sponge, they would be oftentimes sessile. Or planktonic. Planktonic means that they do move around, but you wouldn't call them strong movers or swimmers. They basically drift or weakly swim, such as, say, a jellyfish. So most radial animals are not very strong movers. They're either sessile or planktonic. Bilateral animals often move more actively. They can do everything from run, jump, walk, fly, swim, and have a central nervous system. That's another hallmark of most bilateral animals. And these fundamentally different kinds of symmetry have been around for quite a long time, for at least 550 million years. So by looking at the symmetry and some of the basic body plans of the creature, you can kind of put the creatures into basic groups, and it kind of helps you to sort out some of the diversity that you see in the kingdom of animals. One of the next things we can look at to kind of classify or to categorize different animals is their tissues that make up their bodies. Animal body plans also vary according to the organization of the interior animal's tissues. Tissues are basically collections of specialized cells that work together to do a particular job, and they're usually isolated from other cells by various membranous layers of material. During early development, 3 germ layers give rise to tissues and organs of the animal embryo. Germ layer is basically an early layer of cells that can go on to become a wide variety of different cells, specialized features. We already discussed how one of those germ layers becomes what we call the ectoderm, which is the outside layer. The ectoderm is the germ layer covering the embryo's surface, and this is going to give rise to some of the obvious things that you might think of, like the outer covering of the creature, whether that be skin, feather, hairs, and the like. And also, maybe not quite as obvious, the ectoderm or not so obvious, the ectoderm also begins to become the central nervous system in some phyla of animals. So that doesn't seem as common sense, but it's the outer layer, this ectoderm, in the process of development, which we'll look at vertebrates coming up in another couple of chapters, that actually becomes part of the central nervous system, including the brain and spinal cord. The inside germ layer is called the endoderm. That's the innermost germ layer, and that lines the developing digestive tube called the archenteron. Boy, I'm going to have to check on the pronunciation of that one. So the endoderm becomes basically the tube inside our body, which is the digestive tract leading from the mouth to the anus. This gives rise to the lining of the digestive tract and some of the associated organs, such as the liver and lungs and some of the stomach, small intestine, things of that nature. I checked on my pronunciation there. This is archenteron. Man, having a hard time. Archenteron. So this developing internal tube that comes from the endoderm is called the archenteron, and that's going to develop into usually the digestive tract of most of the animals we're going to look at. As we mentioned, there are some exceptions to all these rules. There are a few animals, some of the very basic animals such as sponges, that don't really have true tissue layers at all. That's how we know for a fact that the sponges are very ancient basal creatures. They lack many of the more distinctive characteristics, the more complex characteristics of most animals. So sponges and a few other creatures that we still call animals that fall into that category, they lack true tissues altogether. So a sponge has no muscle tissue, nervous tissue. It doesn't really have these germ layers that then go on to form more complex parts to its body. If the creature develops from these most basic germ layers, the inner and outer, the ectoderm and the endoderm, we call that diploblastic. Diploblastic, di meaning two, so it has 2 germ layers, basically 2 layers of tissue that make up its body. Diploblastic. These would include the cnidarians, such as jellyfish and sea anemones and comb jellies. So some of your most basic animal creatures are diploblastic. They only have the 2 germ layers. More than most most of the more complex or advanced animals are what we call triploblastic, and this would include humans. Triploblastic animals also have a third layer that develops in between the ectoderm and the endoderm, and this is called the mesoderm. These include all of the bilaterians, there we go, bilaterians, the creatures that have bilateral symmetry. They have a third germ layer called the mesoderm, and that would include everybody from the pretty simple creatures like flatworms all the way up to arthropods, which would be like everything from insects to crustaceans to all your vertebrates, which would include everything from birds to reptiles to mammals. So triploblastic means they have three germ layers. They have the ectoderm on the outside, the mesoderm in the middle, and the endoderm as the very interior germ layer. Whereas we also already discussed that the ectoderm is going to form the outside covering of the animal as well as the central nervous system. We know the endoderm is going to form the digestive tract of the creature. The mesoderm forms the muscles and most other organs that fall between the digestive tract and the outer covering. Another way that we can categorize animals is based on their interior body cavity. Most triploblastic bilaterian animals also possess what we call a body cavity, which is a fluid or kind of air-filled empty space basically between the digestive tract and the outer body wall. So think of that sort of inner mesoderm layer. You have this opening, this body cavity. A true body cavity is called a coelom and is derived from the mesoderm only. So if the body cavity comes from the mesoderm, which is that middle layer of developing cells that we talked about, it's considered a true body cavity called a coelom. Coelomates are animals that possess a true coelom, and that would include humans, mammals, and the like. And then there are other variations on that theme as well. So here is a coelomate. This would be a type of worm that we're looking at here, and it has a true coelom. It has an outer body covering that came from the ectoderm, and then it has a digestive tract in the very middle, the yellow here, that came from the endoderm, and then you see the red. The red is going to be a tissue layer lining the coelom and the suspended internal organs that came from the mesoderm. And then you can see the opening in here. There's going to be some space basically, and it's going to be air or fluid filled, and that opening is called the coelom. So because his coelom came from all surrounded by red, it came from the mesoderm, this would be considered a coelomate, categorized as a true coelomate. There is a variation of this body form called a pseudocoelom or a pseudocoelomate, pseudo meaning false or fake. A pseudocoelom is a body cavity derived from the mesoderm and the endoderm, and triploblastic animals that possess a pseudocoelom are called pseudocoelomates. And so here you have a different type of worm, a different creature, where his opening, the body cavity that you see, is basically formed from both the endoderm and the mesoderm. So you see the opening, the body cavity touches both the red and yellow tissues. That makes it a pseudocoelomate. And you can see the difference between those two, where the opening, the body cavity here, is only touched by the mesoderm. It was formed only from the mesoderm. That's a true coelomate. And the pseudocoelomate has the same opening, but you see it touches both of the tissues. It was derived from both of the tissues, both the endoderm and the mesoderm. So this would be called a pseudocoelomate. Finally, there are a few creatures, and this would usually be some of the more less complex, the more simple ones. There are some triploblastic animals that lack a body cavity altogether, and they're called acoelomates. You tack an A onto most science words, and the A means against or without. So acoelomates have no body cavity, no opening at all. So here's a planarian that we're going to look at a little bit later. He's an acoelomate. He does have three layers, but you notice there's no opening. There's no coelom at all. He has the wall of his digestive cavity that came from the endoderm. He has a tissue-filled region from the mesoderm, and his body covering came from the ectoderm, but there's no open area, so he has no coelom at all. He is triploblastic, but he has no opening, so he's called an acoelomate. So here is all three of them together. So a true coelomate, its opening comes only from the mesoderm. A pseudocoelomate, his opening comes from both the endoderm and the mesoderm, and an acoelomate has no opening, no body cavity at all. So these three formations of tissue can help separate the different types of animals as well. The creatures that have a body cavity, called coelomates and pseudocoelomates, they belong to what we call the same grade. And a grade is kind of a less formal grouping of creatures. It's a group of creatures whose all their members share certain important biological features. So the body cavity is the biological feature we're talking about in this case. So it's not really a scientifically accurate or formal grouping, but we call them of the same grade. It just means they're a less formal grouping, that they share some particular key feature we're looking at. We're going to take a quick break right there, and we're going to talk about one last developmental issue, but I don't want to make this one too long, so let's take a quick break and then we'll come back and finish up with one last way of sort of categorizing animals, and it has to do with their embryonic development. We'll be right back.